afternoon, everybody, and welcome to Lund University, and welcome to the 2015 Crawford Academy Lecture. My name is Daniel Conley. I'm a professor at Lund University in the Department of Geology, and I'll be the moderator for today. We have a really exciting program. First up will be Stefan Bengtson, who's a professor at Stockholm University at the Natural History Museum. He'll talk a bit about the Crawford Foundation, and also then introduce Andy Knoll, our guest speaker. Andy Knoll should give an inspiring talk. We're hoping that he converts all of you to become a geologist or an evolutionary ecologist, so we have large expectations, and, but he should be inspiring. And then we'll have a question and answer period afterwards, a discussion for a period of time. And then afterwards, we'll have a fika, and there'll be coffee and tea and cakes, of course. So with that, I'll turn it over to Stefan. Please.
Hi, welcome everyone to an exciting afternoon and for the second Crawford Academy lecture. We're very happy to see all you students from schools and the university here. And we're also very happy to see members of the Crawford family and the Crawford Foundation here, without which we wouldn't be here at all. A few words about the Crawford Committee, and particularly about Holger Crawford, who was the brilliant industrialist who founded this venture. He was, when he was in the beginning of his career, he was a co-owner of the well-known Tetra Pak company, and that created the embryo of a fortune that he then used to start a company, Gambro, which produces medical equipment for, particularly for dialysis treatment. So if you should, hope not, but if you should run into kidney problems sometime in your life, the Gambro company may well serve or help to save your lives. And the Gambro company has now grown to a worldwide venture. But Holger Crawford was also a visionary, philanthropist, and a great donor. In 1980, he established the Crawford Foundation, which had its main aim to support scientific research, scientific education, and the education of young people. Particularly, he focused on the interplay between science and the arts, and that's still an important focus of the Crawford Foundation today. Also in the same year, in 1980, he made a first donation to the Royal Academy of Sciences to set up what then became known as the Anna-Greta and Holger Crawford Fund, which is now used as the foundation for the Crawford Prize. And you may or may not have heard of the Crawford Prize, but it certainly came with the ambition to cover fields of sciences that Alfred Nobel forgot. And in particular, that would be the fields of mathematics, astronomy, geosciences, and biosciences. And this prize is financially on par with the Nobel Prize, and in fact is there to reward work within these areas that still have not quite come up to the flair of the Nobel Prize, but the Nobel Prize has 80 years head start on it, so just you wait. 80 years from now, you may find that the Crawford Prize is as well known or better known as the Nobel Prize. Even today, it's, as I said, it's on the same par financially. It's even handed out by His Royal Majesty, Monarch of Sweden. And the winners of this award are selected by committees in the Royal Academy of Sciences, more or less the same as with the Nobel Prize. But the Crawford Academy lecture was started only a year ago, and the main reason was that it was felt that there was a need to spread the knowledge of these otherwise sometimes forgotten sciences now covered by the Crafoord Prize. And it started out with a big bang with the astronomer Sir Martin Rees from Cambridge University, who gave a talk to a group like you, and perhaps some of you were even present then. And he talked about the emergence of our complex cosmos. And this year's topic is in geosciences. Well, I almost said biosciences because it's really geobiosciences, and deals with the emergence of our planet Earth and particularly the evolution of Earth in connection with its thin veneer of life, the biosphere. And our guide through this, through this field, will be Professor Andrew Knoll from Harvard University in the US. And Andy is not only the perfect guide for us through this magnificent period of time or of Earth history, and to guide us through the complex landscape of Earth and life interactions through time. He has even been a major player in shaping this landscape, or at least shaping our understanding of it. Andy holds both geology and biology positions at Harvard, which is fitting because, again, as I said, it's a geobioscience field. And his work extends from the beginning of life on Earth through its ups and downs to mass extinctions in the past and even, sadly, mass extinction of the present. His field studies have taken him to a number of more or less exotic places like Arctic Siberia, Spitsbergen, Greenland, Australia, and Namibia, etc., etc. And is a paleontologist who is then a scientist who studies the evolution of life through clues from the fossil rock record. But he's not your ordinary bone hunter. His material and methods are much more challenging, not only because he's covering a time span of 4 billion years, 4,000 million years, but also through most of that time life was microscopic. And you have to deal with life forms that are invisible to the naked eye, deal with subtle clues in the rocks, with chemical processes in the rock, with the distribution of isotopes in the rocks. And the questions Andy's asking in his research are really fundamental. How was life established on Earth? How does Earth influence life and vice versa? What role does free oxygen play? What triggers evolution? What causes mass extinctions? Etc., etc. And the most important question of them all, are we alone in the universe? The latter question is no longer just for philosophers and fans of Star Wars, but it can be addressed scientifically within the radiant field of astrobiology. We may think that we cannot yet observe life on other planets, but Earth a couple of billion years ago was actually a very exotic planet, very much different from what it is today. So it's a very good start by studying Earth. And just like astronomers, paleontologists can see through deep time when they study their distant objects, whether they be distant in space or in time. But we have not only Earth to take our clues from in astrobiology. When I listed some exotic places where Andy Knoll had been doing fieldwork, I omitted the planet Mars. And if you ask him kindly after the talk, I haven't talked to you about that, but Andy may just tell you what it's like to drive a rover vehicle on the red planet. So to come back to Holger Crawford and his early years with the Tetra Pak company, tomorrow when you have your breakfast, give a thought of gratitude to the milk package in front of you. Not just because without it you would have milk spilled all over the table, but because that package seeded the funding that made it possible for you to come here to meet one of the foremost geobiologists of our times. So I give you Andy Knoll, and Andy, please, the floor and the audience are yours.
Well, good afternoon, everyone. Let me begin first by thanking Stefan for that kind introduction, and also thanking the Crawford family, the Crawford Foundation, and the Swedish Academy of Sciences for the much appreciated invitation to be with you. And I have to say that as someone who spent a career in science, it's a particular pleasure to talk to an audience that includes so many of the faces of the scientists of tomorrow. What I want to talk to you about today, in some ways, is a question of history. But it's not the conventional history that you learn about in class. That is, we're not going to talk about World War II and its aftermath, or Gustavus Adolphus, or the dawn of the Renaissance. The history that I want to talk about, as Stefan already alluded, is one that is global in scope and 4 billion years in duration. It's the history of our planet and the life it supports. And what I want to do, in part, is answer one question, and that is, how can we reconstruct the state of the Earth, that is, the nature of life and environments, as they existed 1 billion or more years ago? And hopefully I'll convince you that while there are uncertainties remaining, a lot of them, that in fact we've come a long way in being able to reconstruct the sweep of evolutionary history throughout the history of our planet and the accompanying development and change in the nature of Earth's surface environments. And in many ways, the argument I really want to make is that Earth and life have been closely intertwined for the entire 4 billion years of Earth history. And I think that's an important argument to make because the general form of that argument is that many of the most exciting questions in science today fall in the gaps between, or at least at the interface of traditional disciplines. So while the Crawford Prize has separate prizes for geosciences and biology, one of my arguments is that from my perspective at least, life science and earth science are different sides of the same coin. I don't think I can understand the history of life without interpreting that in the context of Earth's dynamic environmental history. And likewise, I can't explain environmental history without knowing how organisms have contributed to the development and maintenance of environments. So the broad argument then is that Earth and the life it supports are not separate entities that should be studied by separate disciplines, but in fact, Earth and life are closely intertwined and cannot be separated. Oops, there we go. So I'm going to actually begin with just 3 slides, and let me step out here so I can see the slides too. But I want to start out with 3 slides of intellectual history that I think will set the stage for the comments that follow. And I'm going to start at the beginning of the geological venture. This person that you see here is James Hutton, late 18th century son of the Edinburgh, Scotland Enlightenment, and generally considered the father of geology. And that title is accorded to him largely because of the volume you see on the right, which he first wrote in 1788 called A Theory of the Earth. And what's interesting about that volume is that if you actually read it, you find that Hutton's motivation was explicitly biological. Like many other late 18th century naturalists, he would go up into the hills and mountains north of Edinburgh, and he was very impressed by the goodness of fit between the shrubs and trees that lived on the mountainsides and the environments that they inhabited. By the same token, he would go down to the bay north of Edinburgh, the Firth of Forth, and he saw that the sea anemones and seaweeds that were living in the bay showed a goodness of fit between their biology and the environments they inhabited. But Hutton saw something else that his contemporaries didn't see. He saw that weathering and erosion were slowly but inexorably destroying the mountains that provided a habitat for those trees and shrubs. And he knew that the erosional products, that is sand and mud, were coming down rivers and slowly filling in the bay. And so the question that motivated Hutton was, how can species persist on the Earth for long intervals of time when the environments to which they are fit are in a constant state of decay? And Hutton's answer, which was a brilliant one, was that the environment is continuing, but it's actually continuing in a dynamic fashion. That is, any given mountain will through time erode and disappear, but some process, and he thought it was heat in the interior of the Earth, will rise up new mountains so that at all times there will be environments that are fit for the plants and animals that live on mountains. Well, let's move forward then by about 70 years or so and visit someone who's probably much better known to you, Charles Darwin. And in his famous book, The Origin of Species in 1859, Darwin argued that populations are just as dynamic as the Earth system described by Hutton. That is, species are not constant, that populations through natural selection are continually adapting to this dynamic environmental landscape. So both Earth and life are subject to change. And then finally, a less conventional choice. This is Percival Lowell, who was a very famous American astronomer at the end of the last century. And when he's spoken of today, he's often spoken of in ridicule for some books that he wrote, in particular a series of books on Mars. And they were predicated on this map of Mars that he drew that you see in the upper right-hand corner. And what you'll see here are all these straight lines covering the surface of Mars. Lowell thought that he could actually see those through his telescope, and he interpreted them as canals that were built by a technologically advanced civilization to take water from seasonally melting ice caps on the poles to parched cities in the equatorial regions of a very dry Mars. Now, even at the time he wrote that, most astronomers were not very impressed by that idea. On the other hand, if you actually read his books, Lowell makes some very modern-sounding arguments in the sense that he argued that Mars was once a wet planet and has dried out through time. And for very different reasons, we actually think that's true today. This is a picture of Mars taken a few years ago by the NASA rover Spirit. And there's no question that today, no life that we understand could live on the surface of this planet, let alone technologically advanced civilizations. But early in Mars history, there were rivers, there were lakes. And Lowell thought that planets not only change through time, but change directionally. And so if you take Hutton's ideas, those of Darwin and those of Lowell together, you realize that not only can environments change through time in a directional sense, and I'll argue that the Earth has changed in exactly that way, but since both life and environments can change, that there opens up the possibility of coevolution between Earth and life. The changes in environments will make new types of evolution possible. New evolutionary innovations will have an influence on environments. So that's the argument, and it's entirely predicated on our ability to actually reconstruct the history of the Earth. In order to do that, we need libraries that actually preserve a record of Earth history, and those are all around us. So what you're seeing here are sedimentary rocks that are exposed in Western Australia. This is a group of rocks I've worked on a number of times over the years. And by the study of those rocks, I can with some confidence say that 1,870 million years ago, nearly 2 billion years ago, in this part of the world, there were coastal environments that were covered by microbial communities living beneath an atmosphere without much oxygen. Now you might say, well, how can you actually say that? And the reason I can say it is because time, life, and environment all leave an interpretable record in sedimentary rocks, laid down one layer after another through time. So time we tell because of the phenomenon of radioactive decay. That is, most elements have radioactive forms that actually decay through time to some stable daughter product. We can actually measure in the laboratory the rates at which those isotopes decay. And if we have the right minerals, we can then use that principle to actually to date old rocks. The mineral that we use is something called zircon. It's a zirconium silicate mineral. And what's important about it is that its crystal lattice is such that you can actually stick a little bit of uranium into it, but lead won't fit. Now, the reason that's important is that at least 2 different isotopes of uranium decay through time to produce lead. So when a chemist goes and looks at the amount of uranium and lead in that rock, you can actually convert that into a date. Fortunately, there are volcanic rocks in this sequence. They have minerals in them which can be dated, and that allows us to say with some reasonable resolution what the age of these rocks are. So what can we say about environments? And this is just a snapshot of that cliff we were seeing, and you'll see a couple of things. There are these bowed up layers that I'll get to in a minute. But notice that there are some very light rocks that looks like they're almost squeezed in among these laminated sediments. These are all calcium carbonate or carbonate rocks. And if you look up here, you'll see that these lower beds have actually been truncated and then covered by more sediments. So what that tells us is that these sediments must have been formed close to sea level because they're left high and dry every now and again. They get eroded, and then we get more sedimentation. And in fact, these white rocks that seem to almost be squeezed into the older rocks are something that geologists call beach rock, which form today in carbonate precipitating environments when those environments are actually exposed to the atmosphere. So the general form of the argument is that there are physical and chemical features of sedimentary rocks that enable us to place these rocks in space as well as in time. We get some sense of biology because it turns out that we understand how these domes form. And they actually form by the interaction between microbial communities and the physical environment. And in other parts of this succession and in other places of the same age, we actually can put some of these rocks under the microscope and see these very small, sort of 1 to 10 micron scale microfossils of bacteria. There are no bones, no shells, nothing like that, no sense of animals in rocks of this age, and there won't be any record of animals for more than 1 billion years after this. But bacteria have actually left a calling card of their existence. Now, one of the things about the rock record is it is a record of pattern. What we would like to know in many cases is processes at work when the rocks were laid down. We would like to know the state of the environment when the rocks were laid down. But all we have is pattern, chemical and physical pattern. And so we can convert that into an understanding of process by looking at things that are going on today. So for those domed rocks called stromatolites, it turns out there are a few places on Earth today that you can go and still see stromatolites forming today. This is a place called Shark Bay in Western Australia. It is to people in my field almost what Mecca is to a good Muslim. It's the place you go to really find out how things work. If you cut these domes in half, you see they have these bowed up laminations like we see in the geologic record. And it's really the studying of the microbiology and how it interacts with physical processes today that allows us to make these inferences about the past. Well, we get more inferences from the chemistry of rocks. It turns out individually bacteria are tiny, but there are so many bacteria in the oceans that they actually influence the chemistry of the oceans in a way that can be recorded in rocks. So what this little cartoon is meant to show, that when organisms fix carbon dioxide into organic matter in photosynthesis, they actually do it in a way that leaves a geological signature. That is, if we look at the CO2 that's either in the atmosphere or dissolved in seawater, some of the carbon, about 99% of the carbon in that CO2, is the lighter isotope of carbon, carbon-12. 6 protons and 6 neutrons. And about 1% is carbon-13, which has an extra neutron. When these rocks formed, there was also about a couple parts per trillion carbon-14, which is radioactive, but that decays away over thousands of years and doesn't impact the study of the early Earth. The reason it's worth mentioning these isotopes is that when a plant or an alga or a cyanobacterium fixes CO2 during photosynthesis, it actually favors the lighter carbon dioxide with the lighter isotope C12. And so if you take a mass spectrometer, you can actually measure the amount of C12 and C13 in limestones that are forming from seawater, and you can measure the amount of C12 and C13 in organic matter that's preserved with those, and they differ by about 28 parts per thousand, which is the signature of Rubisco-based photosynthesis. So the general point is we can actually use the chemistry of these rocks to say something about the functioning of the carbon cycle in our planet's deep past. I could have told the same story for sulfur by looking at sulfur in minerals like gypsum or iron pyrite, called fool's gold in English at least. You also see an isotopic calling card that allows us to reconstruct a biological sulfur cycle through Earth history. And finally, chemistry can actually help us to understand environments. And once again, what you see in the background here is just a satellite image of the Black Sea. And as some of you may know, the Black Sea is interesting in that it has an oxygenated surface layer, but at depth, there is no oxygen. And it turns out, if you look at a seemingly mundane feature of muds, which is the iron that they contain, in the modern, you can actually cleanly distinguish between sediments that are forming beneath seawater that has oxygen in it and sediments that are forming beneath seawater that doesn't contain oxygen. So in this diagram here from a paper we wrote some time ago, this ratio here, which is highly reactive iron over total iron, what that really means is highly reactive iron is iron that's present in oxides or carbonates or sulfides. That is iron that is forming in the surface environment and can be easily manipulated. And then the rest of the iron is iron in rock-forming minerals. If you look at muds accumulating beneath shallow waters of the Black Sea today, they have a very low ratio of this. If you look in muds that are accumulating in the deep Black Sea, it's much higher. Also, on the x-axis here, there's something called DOP, which means degree of pyritization. And what that really says is, of all of this highly reactive iron, how much of it is pyrite? And again, when there's oxygen in the water column above the muds, that tends to be low, and when there's not, it tends to be high. So what we get from modern environments is this distinction between oxygenated environments where these factors fall in here and non-oxygenated environments. So what we did was went back to rocks that were about 1,800 million years old, and those are the squares and triangles here. It's a little bit difficult to differentiate them in this black and white column, but the important point is that when you went at depth in oceans from long ago, they look like the deep Black Sea. And in fact, everywhere you go in rocks of this age, the chemical record tells us that the oceans look very different from today and that there was probably much less oxygen in the ocean atmosphere system than there is today. Okay, so that's what we're going to do. We're going to look at the physical nature of the rocks. We're going to look at biological records. We're going to look at chemical records. And we're going to start out by going to about the oldest rocks that we can look at profitably, which are sedimentary rocks laid down 3.5 billion years ago in what's now Western Australia. And I like this picture, which was actually taken on an expedition there, because it shows that in the field there's 3 types of earth scientists. There are, on the one hand, when the car breaks down, there are those who fix it. Second is, of course, those who watch those who are fixing it. And of course, there are those of us who just get our cameras and take up pictures of the other two. If you go to these rocks, mostly it's volcanic rocks, but there's thin successions of carbonate rocks, which you see here. And if you get your eyes in, you're going to see that you have laminations that here are building up into a cone, somewhat like we saw in that previous slide. And in fact, these are 3.5 billion-year-old stromatolites. And in particular, their microscopic features convince me at least that these record the presence of microbial communities on the Earth's surface 3.5 billion years ago. We can also look at, carbon isotopes of these rocks, and they tell us that 3.5 billion years ago there was a biological carbon cycle, most likely driven by photosynthesis. And indeed, there are fragments of materials that go back to more than 4 billion years ago that already contain a carbon isotopic signature suggestive of carbon fixation by organisms. So the punchline there is that not only is Earth a biological planet, perhaps uniquely in our solar system, at least at present, Earth has been a biological planet for most of its history. We can also look at sulfur isotopes in minerals, and they suggest there was also a biological sulfur cycle going on at this time. Now, there's one other thing we can say about this world, and that's that there was essentially no oxygen in it. If you would be transported back to this world, you would asphyxiate in a few minutes. So how do we know that? How can we make a statement about the amount of oxygen in the atmosphere 3.5 billion years ago? And again, we do it because of chemical pattern that we can interpret in terms of state and process. So one of the most amazing and wonderful rock types that we see in early rocks is something that we see here. And these are actually 2.5 billion-year-old rocks from Australia. But this is a rock type called iron formation. Much of the iron used by technology since the industrial age comes from sedimentary rocks that were laid down in the first 2 billion years of our planet's history. These kind of rocks cannot, in principle, form in today's oceans. And the reason is that in order for them to form, iron has to be able to travel in a dissolved state through seawater. And when iron comes in contact with oxygen, it almost instantaneously will oxidize to a product that will be precipitated out and will no longer be soluble in seawater. So these rock formations are testimony to the fact that there was not much oxygen in the oceans. Also, if you look at sandstones that were deposited at these times, you'll sometimes find things like this. This is a large sand grain, and the sort of shiny yellow stuff that you see there is pyrite, iron sulfide. Now, we know that if you put pyrite outside in the environment, it will start to oxidize to sulfate very, very quickly. So when you see this kind of unstable mineral, at least in the presence of oxygen, in these sandstones, that tells us that that pyrite was exposed at the Earth's surface in some sort of mountainside, was eroded and carried down through rivers, and then deposited in deltas without ever coming into contact with much oxygen. And in fact, there's a number of oxygen-sensitive minerals that accumulate as sand grains on the early Earth that you don't find again today. There are iron carbonates, uranium minerals, as well as these iron sulfides. And then, perhaps most quantitatively and most unexpectedly, there was a discovery made by James Farquhar, a geochemist in the United States. By looking at rare isotopes of sulfur, he saw this amazing pattern through time. This is just Earth history from today back through 4 billion years. And you'll notice that the left-hand side and the right-hand side of this curve are distinctly different. And that difference relates to the relative amount of the rare isotopes of sulfur in sedimentary rocks. And it turns out that this kind of chemistry, without going into it in detail, is a chemistry that will only form on the Earth's surface when you have photochemistry, that is the influence of solar radiation on the atmosphere in the absence of oxygen. So that tells us that the first 2 billion years of Earth history were very different from the last 2.5 billion years. Okay, so we're living a world with microbes, but without oxygen, probably for at least 1 billion years of Earth's early history. Now, what would a world like that have been like? We tend to think of photosynthesis in terms of green plants or algae, or if you're really well educated, cyanobacteria, which are a type of photosynthetic bacteria. And those organisms famously take CO2 and water and convert that into organic matter and give off oxygen as a byproduct. But if you look around today, there are at least 5 different other groups of bacteria that can do photosynthesis, but do not use water as a source of electrons for photosynthesis, and so do not give off oxygen. And it's very likely that photosynthesis on the early Earth was that kind of photosynthesis that seems so exotic in our oxygen-rich world today. Don Cantfield, a very well-known geologist in Denmark, actually tried to quantify the amount of primary production or photosynthesis you could have on this early Earth before you had a cyanobacteria. And the numbers he came up with are pretty reasonable, I think. He says that the major source of electrons for photosynthesis before cyanobacteria would have been iron. And the amount of iron that would have been available in the oceans would have allowed photosynthesis at levels maybe 10% or so of today's. So in terms of carbon production, the world's a little bit different on the early Earth than today. Well, what happens to complete the carbon cycle? I mean, in this room, we are all completing the carbon cycle. Through respiration, you're taking organic matter, reacting it with oxygen, and returning CO2 and water to the environment. How did that work before there was oxygen? Well, this is a diagram from Ken Nielsen, a microbiologist in the United States. And all it really says is if you take fuels and things that can oxidize those fuels, this is just telling us it's something called electron potential. But what it really says, if you want to have a reaction between something on this side of the graph and something on this side of the graph, any reaction where you draw the line downward from one side to the other will actually yield energy that organisms can use. So Ken has drawn a happy bacterium here. If the reaction connects in this direction, it actually requires energy to run, so organisms will not use that as heterotrophs. Now, if there wasn't any oxygen, how would this work? Well, it turns out if there's no oxygen, you can't have respiration the way that you and I are doing now. And not only that, if there's no oxygen, there won't be much oxidized nitrogen, nitrate or nitrite, and there won't be much oxidized sulfate. Those things can be used in respiration by bacteria today, but they would have been in fairly low supply. So on the early Earth, we turn back to iron. And it may be that iron was the principal way that organisms respiring would complete the carbon cycle. So the suggestion then is that on the early Earth, the iron cycle and the carbon cycle may have been closely linked in a way that they no longer are. And I think that in detail, the physical and chemical features of these ancient iron formations actually support that view. So what changes? Why is it that after 2 billion years, the amount of oxygen starts to increase in atmospheric history? Well, we know the source. There's only one source of oxygen on this planet that is large enough to actually provide the oxygen that we see in the atmosphere today, and that is oxygenic photosynthesis. So what looks like scum to you are actually the heroes of the revolution in Earth history. Cyanobacteria are the source of the oxygen that makes our planet habitable to you and me. And in fact, the same record that we used earlier to say that there was no oxygen on the early Earth actually can be used to say that about 2.4 billion years ago, the world changed and essentially oxygen began to accumulate on a permanent basis in our planet. And that's going to make a lot of new kinds of biology possible. Now, people still argue about why that happened. Basically, somehow you have to change the balance between the production of oxygen by photosynthesis and the consumption of oxygen by the oxidation of either minerals or organic matter. And there's still no real agreement on it. It's an opportunity for the next generation of scientists. But in general, it appears to relate to just the physical evolution of the Earth, which rebalances the supply of nutrients and things that will consume oxygen. Well, however it happened, we can ask an interesting question. How much oxygen appeared? Did this oxygen revolution 2.4 billion years ago create a world like today, or was it some intermediate state? And there's pretty much consensus now that at least qualitatively, the world that began 2.4 billion years ago was still very different from our own. This is a diagram taken from a paper by Tim Lyons, a prominent geochemist. And what it says is that early in Earth history, there's no oxygen, even though there may have been transient oases where early cyanobacteria were oxygenating the water column. It also says in the last 500 million years, a world which is populated by large and active animals, there are oxygen levels at least broadly similar to today's. But between them, for 2 billion years of what's called the Proterozoic eon of Earth history, there was some oxygen, but not much. Maybe 1 to a few % of today's oxygen levels. Now, the question is, how do we know that? And again, we know it by looking in detail at the chemistry of oxygen-sensitive minerals and their isotopes. And then the question is, what are the biological consequences of that? Well, it turns out if we go back to this Nielsen diagram, once we make oxygen, we can then make nitrate, which is important as a fertilizer as well as an oxidant. We can make sulfate. And indeed, the richness of the biological constituents of the carbon cycle increases dramatically once oxygen is present. Also, for geologic reasons, the quality of the geologic and in fact, the fossil record increases markedly. So once we get into the Proterozoic, there are abundant microfossils. All of these are actual body fossils of cyanobacteria. This would be about 15 microns across. Each of these is 4 microns. Tiny little fossils, but preserved beautifully in rocks and still available to us today. And stromatolites are everywhere. I love this picture. It was taken in northern Siberia. In part, I love it because my colleague Misha Semikhatov here is a perfect field partner because with his hat on, he's precisely 2 meters tall. So you can actually calibrate your photos by having Misha in them. Now, if you're looking at this picture, you might say, well, I see this little stromatolitic reef. And you're absolutely right. That is a patch reef made by microbial communities, just the way corals would make a reef today. It would be a reef about the size of this table in the front of the room. So if you got that right, give yourself 5 points. But it turns out this is a 15-point question. If you look at the surface that Misha is standing on, that's also a microbial reef, but it's a reef about the size of this room. And if you look at the wall behind him, you can faintly see these vertical structures here. That's a reef that's larger than this building. In fact, in Earth's middle age, in the Proterozoic eon, we see reefs built by microorganisms that have the same dimensions as coral reefs today. We also have carbon and sulfur isotopic signatures that tell us about the continuing function of the carbon and sulfur cycles. And we actually have, there are some molecules that resist decay and just like body fossils can be associated with organisms. And so we know that at this time, most of the primary production in the oceans was done by these cyanobacteria. So it's still halfway through our planet's known biological history, very much a microbial world. But there's a new component of this microbial world, and that's eukaryotic cells. And on the left here, you see a little diagram from a paper some years ago by Christian de Duve, and it tells you things that most of you already know about the eukaryotic cell. Unlike bacteria, which have a single circular chromosome that is not compartmentalized relative to the rest of the cell, in eukaryotes, you have multiple linear chromosomes that are contained within a membrane-bounded compartment called the nucleus. Also, there are organelles where important features of metabolism take place. So respiration takes place in mitochondria, in photosynthetic cells. Photosynthesis takes place in chloroplasts. And as some of you might know, the chloroplast in an oak tree is a lineal descendant of a once free-living cyanobacterium that was swallowed by an early protozoan and eventually reduced to metabolic slavery. So eukaryotic cells are committees in a sense. The mitochondrion was once a free-living bacterium as well. Also, the thing that really makes eukaryotes special, and what I think is actually responsible for much of their ecological and evolutionary success, is that they organize the interior of the cell by means of molecular filaments called a cytoskeleton, and they have a very dynamic membrane system, which allows eukaryotes to do something that bacteria and archaea cannot, and that is a eukaryote can actually engulf and swallow particles. So now predation becomes possible. And already in rocks from Earth's middle age, about 15 or 1600 million years, we see cells whose morphological features are known from eukaryotic cells today, but are not known from bacteria. Sadly, we don't have billion-year-old DNA. We don't have billion-year-old cytoskeletons preserved. So all we can look at is the physical and chemical features of these rocks, but they tell us that in Earth's middle age, eukaryotes were part of ecosystems. That said, eukaryotes appear to be relatively minor parts of ecosystems for perhaps the first half of their existence. We can go down to 800 or 700 million years ago before the chemical record suggests that eukaryotic algae are becoming important primary producers. And in rocks, roughly speaking, around 800 million years old, we start to see a much increased diversity of eukaryotic organisms in the fossil record. So one can ask, why is it that so long after they first appear, eukaryotes arise to a new level of diversity and ecological importance? And I'd just like to float an idea, and I will caution you that this is an idea that I like very much, but is not consensus, so take it as opinion rather than established fact. But I've been very struck that when we look at the record of the early divergence of animals, around 550 to 500 million years ago, that people have made very strong arguments that the advent of carnivory, animals eating other animals, is an important ecological driver of early animal diversification. So I've been thinking, what would happen if midway through the evolution of eukaryotes, we had the introduction of eukaryotes that ate other eukaryotes? And I think the ecological expectation is that that would lead to a diversification of eukaryotes. So how can we, if that's a viable idea, how do we actually think about it? Well, first of all, we can go to the fossil record. And for the first time in rocks of this age, we see fossils of protozoans and algae that were actually armored. So what you see in the top here is a so-called testate amoeba. Two different groups of protozoans have evolved this. This is basically an amoeba. You can see its pseudopod here, but it's an amoeba in a bottle. It has its own test. This is a modern testate amoeba test. This and these come from 750 million year old rocks in the Grand Canyon in the United States. Very similar in size, shape, and organization. And indeed, this one from the Grand Canyon is very similar to this, a modern testate amoebozoan, which eats other eukaryotes. So you can take 3 arguments from these fossils, which appear globally between 800 and 750 million years ago. First of all, at least some of them appear to be related to modern groups of protozoans that eat other eukaryotes. Second, they have armor. And one wants to say, what's the functional reason to have armor on this early Earth? And avoiding getting eaten is a good reason to have armor. And then there are these things that my former student, Susanna Porter, noticed when she was working on these, little half-moons that are cut into some of these shells. And it turns out that's not just damage by mineral growth. There's reason to believe that that's a biological signal. Something was actually breaking into and preying on these cells. So we get arguments from that. The other thing that's become abundantly known recently are these things. These are scales. Many groups of eukaryotes today are covered in scales, again, in many cases as armoring. And my former student, Phoebe Cohen, has looked at these carefully in some 800-million-year-old rocks from northwestern Canada, and she finds about 40 different kinds of scales. That's 10 microns, so these are exquisitely beautiful scales. And this is, in fact, the most diverse record of eukaryotic life before the advent of animals. We know from today that many different groups of protozoans and algae make scales. So these are little siliceous scales that are made by a group called the haptophytes today. This one from the work of Willi Feusner in Austria is amazing. This is a ciliate, like a paramecium that you've probably seen in biology class, and it's completely covered by these scales. So that shows us that functionally we can draw some lessons from today for these fossils that appear 800 million years ago. We can also go at this from comparative biology. We can look around and see what do eukaryotic organisms actually eat and how does that relate to their phylogeny or family tree. So working with a Brazilian protostologist named Dan Lahr, we've been doing this kind of fun, although complicated exercise of looking at phylogenies, just evolutionary relationships based on molecular comparisons among eukaryotes. We just look at the tips of these, which are individual species, and determine their feeding mode, which we see here. And then there are algorithms by which you can use this data to develop a sense of the probability of what was the ancestral trait. And when you do that, particularly for the most likely root of the eukaryotic tree, which is here, it suggests that early eukaryotes actually fed by eating bacteria-like structures, bacteria-sized structures. Then some of them became photosynthetic by eating a chloroplast. Some of them became osmotrophic, which is like fungi that just absorb molecules. And some of them, the ones in red, evolved the ability to actually eat other large cells. And it turns out that when you look at these on a so-called molecular clock, which tries to estimate the time of occurrence of these evolutionary divergences, it suggests that the major groups of eukaryotes that eat other eukaryotes are becoming important, around 800 million years ago, just when you'd hope they might. Finally, one other one, and then we'll sort of draw this to the last arguments, is that with some colleagues in Italy, Mario Giordano and his students, we've actually been doing experiments to see what would be the consequences for individual organisms of living in a world with or without predators. And so what this slide just shows is you take a green alga or a cyanobacterium, and the white line is growth rates when you're growing them only in pure culture, only by themselves. And then for different levels of sulfate availability, and we did that because the amount of sulfate in the oceans has changed systematically through time, we first grew them by themselves, then in the presence of a ciliate predator, and then in the presence of a little arthropod, a copepod predator. And what you can see for the green algae, except for this one treatment, when you add a predator, the growth rates increase markedly. And in fact, The palatability and nutritive value increases. The amount of protein actually goes up substantially when you have predators in the environment. Cyanobacteria don't seem to notice. So what that suggests is that when you add predators, you might actually tilt the world of photosynthesis in a way that would favor the rise to dominance of eukaryotic photoautotrophs. And in fact, the fossil record suggests that between 8 and 600 million years ago, green algae become important members of the phytoplankton for the first time. So eukaryotes are slowly becoming ecologically dominant in the world. So, last argument on this, which is molecular clocks suggest that animals first started to differentiate on the same time scale that we've been talking about, something like 800 million years ago. And might that rise of animal multicellularity somehow be related to this new world with eukaryotic predators in it? There are also a lot of algal groups that are evolving simple multicellularity in new ways at this time. And we get some insights to that from some really nifty experiments that were done some years ago by Boras et al. And what they did was they grew single-celled green algae in a flask, and those things will repeat their single-celled life cycle forever, basically, if you leave them in this laboratory condition. And then they introduced a protozoan predator. And as it says here, within 10 to 20 generations, all the green algae were multicellular. They all had anywhere between 8 and 16 cells. And the reason is those green algae that stick together after they divide can't be eaten by the predator. That becoming multicellular is actually a very effective defense against predation by a single cell that's trying to gobble you up. So it may be that the advent of simple multicellularity in a number of groups, including the group that would give rise to animals, is related to the new ecology of the world that began around 800 million years ago. Well, finally, we start to come closer to a recognizable world. Even though it's thought that animals first differentiated around 800 million years ago, we don't see a record of macroscopic or large organisms thought to be animals until about 560 million years ago. And one can ask, why is it that there is this, essentially a quarter of suspected animal history goes by before we actually see large animals in the fossil record? And what we might do is go back and say, well, when do we start seeing oxygen levels that are comparable to today's? And it turns out that the second major oxygen transition is happening in this same time frame. Now, there's a lot of work going on, again, as to why that should happen and what the actual course of oxygen change is, because it looks to have been kind of a bumpy road for a while there. But it is the case that oxygen would have an influence on our own group, the animals, as well. So we can ask the question, how much oxygen do you actually need for an animal to live? And an interesting answer to that was provided a few years ago by some experiments in Denmark by Dan Mills and Don Canfield and their colleagues, who grew a simple sponge at very low oxygen levels. That is, 0.5% to a few percent of oxygen in the oceans today. And the sponges could grow. They could grow without modern levels of oxygen. Now, again, 1% of oxygen would kill you in a couple minutes. So it isn't that all animals can grow at this. But simple animals can grow at low oxygen tensions. We also get some work some insights from ecology. My former postdoc, Eric Sperling, now at Stanford University, did a nice study looking at ecological data, because today you can go along gradients in the oceans where you go from full oxygen down to no oxygen. And ecologists have done a good job of looking at the ecology along that gradient. And it turns out that at very low oxygen, that is 1% or so of today's oxygen levels, you still have animals. but they're 100 microns long and 10 microns wide. There's very low diversity, and as you go up to something like 8 or 10% of today's levels, you actually increase things like diversity and something I'll get to in a minute, the presence of carnivores, in a way that by around 10% or so of present-day oxygen levels, you at least have a world that's broadly tolerable by animals. And that's kind of cool because we come back to this ecological argument about carnivory. If carnivory is important to the evolution of animals, when does carnivory become an important process? And here, it's kind of fun because for decades there have been two groups of scientists, half of them saying oxygen is what made animals possible, half of them are saying ecology is what made animals possible. But it turns out, again, those are two sides of the same coin, because at very low oxygen levels, you can have simple animals, but you need more oxygen, perhaps 5 to 10% of today's levels, to have carnivory. And so what we suspect is that Earth may have transited across that threshold, ecological threshold, between 600 and 550 million years ago. And once then we get stably high levels of oxygen, the modern world of animal and plant biology becomes possible. And that makes possible the modern world. This might not seem modern to you, but to me it looks very modern. These are all Cambrian rocks. There are trilobites. This, actually from the work of Stefan Bengtson, is a mollusk. There are annelid worms. There are vertebrates. There are echinoderms. And there are things that kind of wish they were arthropods and aren't quite yet. So we now see this world of large animals, and that will continue to the present. So how do we conclude then? I hope to have convinced you that our Earth has a very, very deep and ancient biological heritage, that biology has changed throughout Earth history, that environments have changed throughout Earth history, and as often as not, environments and life are changing on comparable timescales that suggest that each is evolved in the course of the other. And then the sort of operational conclusion is that we actually learn about the past not by being biologists or by being geologists, but by gathering data from comparative biology of living organisms, from the study of modern environments, and from both the paleobiological and geochemical study of the early Earth. And the postscript is I stopped my narrative just as the familiar world of animals is taking place, but I don't want you to give you the impression that environments have been constant through the last 500 million years. In fact, they're not. We know from plate tectonics that the continents are constantly shifting. This is the world as it existed 250 million years ago, very different from today. We know that climate changes episodically. This is a photograph of the world as it existed 20,000 years ago at the height of the last ice age, somewhat different from today. By the use of geochemistry and modeling, we believe that both oxygen and carbon dioxide levels have changed through time. And I love this depiction. It's a depiction of the last day of the Cretaceous period. And here's two dinosaurs on what's sadly going to be their last date. And they're looking at this phenomenon in the sky, which is actually an 11-kilometer meteorite hitting the planet, which caused mass extinction. So the shape of biology has continually been changing, molded in part by the dynamic Earth system. And I leave you then with this slide from the work of Jim Barry in the United States, which shows not the amount of CO2 in the atmosphere over the last 400,000 years, but the rate of change of CO2 through time. And here you go through the last ice age, and then you can see that with the beginning of the industrial age, it gets pretty interesting. Here you see the rate of pH change in the oceans, and you see the same thing. And the important point is we are actually entering a period of environmental change that is notable on a geologic timescale, that we are, you, your children, and your grandchildren are going to experience these changes in the physical Earth system that are on a par with things that have only episodically been seen over the last few 100 million years. We know the cause of it, it's us, And in this change, if we have the will and the science and technology to do so, we can actually do something about it. But I think as you go through your lives, it is not only that life in the past is subject to environmental change, but your lives will be as well. So thank you very much. So do I just take questions? So you'll moderate the questions? Yeah.
Excellent. Thank you, Andy, for that very interesting talk. We have 2 microphones, or 1 microphone. 2 microphones. And we'll take questions from the audience, and we'll have a discussion about Andy's talk. So does anyone want to be the first question, Ann? There we go. No? Okay. We have one. Great.
But I wonder, what is the view today of the snowball Earth theory and its effect on multicellularity?
Okay, a very good question. It concerns something commonly called the snowball Earth. I didn't talk a lot about climate, just because I only had an hour to talk about the entirety of Earth history. But some of the most interesting features at both the beginning of the Proterozoic interval and at the end are ice ages of a type very different from we see today. And that is about 2.4 billion years ago, and again perhaps twice at about 720 and 650 million years ago, the Earth experienced ice ages where continental ice sheets reached sea level at the equator. And that's a very cold Earth, a very different one from today. And it's actually been hypothesized that the entire Earth, oceans, continents, was swaddled in a veneer of ice, and that's what's called the snowball Earth. I think that is in some ways still a controversial idea, but I think it's closer to being correct than incorrect in the sense that I don't think we can understand these early ice ages by simply saying it's like the Pleistocene ice age, just bigger. From some work actually that we did many years ago, it's clear that these Proterozoic ice ages impacted the carbon cycle in ways that we simply haven't seen in the modern oceans and in the Pleistocene. So I think that whether or not the entire Earth was covered in a kilometer of ice is controversial, but much of the Earth must have been covered in ice, and that would have important consequences for life. On the one hand, we do see that some groups represented in the fossil record disappear, and actually many years ago I wrote a paper on that extinction with the late Gonzalo Vidal, who was a professor right here at Lund. But we also know that many groups of eukaryotic organisms must have been present before the snowball Earth and are still present today. So that puts limits on the severity of the ecology, simply because we have to account for the survival of many, many dozens of eukaryotic groups, including animals. So the question is, then, could it have been provided a spur to evolution? And that's been suggested. One of the problems is that we don't see much change in animals until about 75 million or 55 million years after the end of the last ice age. So it's a little bit difficult to take something that happened 55 million years ago and suggest that's responsible for things that are happening today. It's been suggested that the snowball Earth was related to an increase in oxygen in the atmosphere, but we really don't see much evidence of oxygen change until many millions of years later. It's been suggested that it might have an effect on animal evolution, and it might, but we really don't see it. So the biggest possible change, I think, is that after a mass extinction or after an event like this major ice age, you would certainly have had a lot of environments where there was very little competition because there were very few organisms that were available to populate the ecosystems. And those are the types of environments in which evolution can happen quickly because you don't have to be all that well adapted to survive and reproduce. So maybe it had that effect. But overall, as you can guess from my response, there's still a lot of controversy of what the long-lasting biological consequences of those glaciations would have been. Anything else?
Where can we see the cyanobacteria today?
They are all over. The question is, where can we see cyanobacteria today? And it turns out that about half of the primary production in the oceans today is still by cyanobacteria. You have to go out to the center of the oceans to see it, and they photosynthesize at fairly low rates. But since the actual aerial extent of this center, so-called gyres of the ocean, is much larger than the continental shelves that we're more familiar with, there's a lot of cyanobacteria there today. You can see cyanobacteria forming mats in a lot of places. I'm sure, Stefan, there's places in Sweden where there are cyanobacterial mats forming today. If you're fortunate and go to Oman or the Caribbean, you can see many places where cyanobacteria are still able to dominate. Cyanobacteria tend to do well when they are not subject to being grazed by animals or being outcompeted by seaweeds. And so if at that place, Shark Bay that I showed you, the reason that the cyanobacteria do so well is it's fairly salty water, and so animals aren't competing for space or grazing them. But really, if there's a forest fire in central Sweden, the first organisms that will recolonize that space, at least as photosynthesizers, are probably going to be cyanobacteria. So they're all around you. You just have to look for them.
In one of your key pictures, there were about the oxygen content over time.
Yeah.
And there are two sort of dramatic steps there.
Yes.
Why did that happen?
Yeah, that's a good question. The standard answer to that is if I knew why it happened, I'd write a paper about it. But yes, I think we've done better so far in showing the pattern of history than necessarily explaining it. But I think the pattern is overall correct. As I said, the way people used to think about this was in terms of the processes that produce or consume oxygen. And if you think about that photosynthetic equation, where you take CO2 plus water yields organic matter and oxygen, you think, oh, that's easy. Photosynthesis produces oxygen. But then, of course, you think what we're all doing in this room right now, which is taking oxygen and organic matter and producing CO2 and water. And so it doesn't matter how much photosynthesis you have if you balance that with respiration and other ways of consuming oxygen. So the one way to tip that balance is when you make organic matter and oxygen, you bury the organic matter in sediments. And then the oxygen has no partner to react with, and it can potentially build up. And we actually have some inferences from chemistry that was true both at the beginning and the end of the Proterozoic. The limitation of that is you can build up oxygen, but then you can consume that oxygen again, and things can go back down. So a lot of the thinking in recent years really requires that we change the Earth system itself. And as I said just quickly at the beginning, generally speaking, the factors that might be most important is the availability of phosphorus to drive a nutrient for primary production, and essentially the amount of reducing materials coming out of the Earth's mantle. As the Earth gets older, the amount of reducing materials is going to go down, and given the chemistry of phosphorus, there's reason to believe that may have gone up through time. So it may actually be that, particularly at the end of the Proterozoic, the actual events that pushed oxygen to higher levels may have been almost trivial, but once it gets to that higher level, it can stabilize there, and it's an alternative stable state, and then, barring catastrophe, remain at that state. So that's a very long-winded way of saying we really don't know, but at least I think we're focusing in on a new set of questions and making new sets of geological observations to test them. Yeah.
In the earlier slide, you showed a picture of Pangaea with like all the tectonic plates.
Yeah.
So my question is, how did the movement of tectonic plates influence or what consequences did the tectonic plate movement have on the evolution of organisms?
Great question. As you can imagine, we can answer that with more confidence for younger intervals of Earth history than older, because the way we can reconstruct Pangaea is largely because as the oceans open, they leave a record of their opening. And the record they leave is that as volcanic rocks form at say, the mid-Atlantic ridge, and then spread from the ridge to make the growing Atlantic Ocean, the iron minerals in those rocks become aligned with the Earth's magnetic field. And it turns out the Earth's magnetic field shifts every couple 100,000 years or so. So if you measure the magnetic orientation of these rocks by the ridge, you'll see North Pole's this way, then it's that way, then it's this way, then it's that way. And you actually have this record of ocean growth. So it's pretty easy then to just turn that backwards and close it and see what the continents were like before the current oceans opened up, and Pangaea is the answer. But as you go back into deeper time, we don't have a deep ocean record. Because of plate tectonics, the deep ocean crust tends to disappear. It gets subducted and goes back to the mantle. So then we have to rely on other features to try and build that record. So having said that, the position of the continents has lots of effects. It can affect the rates of weathering, for example. If you put all the continents near the equator, it turns out that rocks will be weathered faster because it's warmer. There's a relationship between weathering and temperature, and that will actually draw down CO2. So some people think this large ice age that we talked about at the end of the Proterozoic eon was because most of the continents were at the equator. More generally, as continents disperse, they will separate organisms that once lived together, and that will affect the evolution of diversity. So today you have ostriches in South Africa, but their close relatives, rheas in South America and emus in Australia. All of those are descended from populations that became isolated by plate tectonics. And plate tectonics really changes climate. It's thought that one of the things that drove us into really the current ice age was first the opening up of the Southern Ocean about 55 million years ago, which dramatically changed ocean circulation in a way that would cool the continents. And then also, beginning about 10 million years ago, the circulation of seawater between North and South America was stopped by the growth of the Isthmus of Panama, and that dramatically changed seawater circulation. So plate tectonics has this very important influence on all aspects of biology, from the altitude, the amount of mountains you have on continents, which has direct effects as well as climatic effects, to a profound influence on climate and a profound influence on the distribution of populations. Yes. I can't, I have this bright light, so if you have a question, you might just yell.
Yeah. Okay, here.
It's over there.
Yeah.
Oh, okay, yeah, I do have this bright light. Yeah.
Yeah, this might be a very hard question to answer, but do you believe that the answer to how we're going to cope with climate change can be found in Earth's history?
That's a very good question. The question is there a lesson from Earth history for our future? Maybe just to generalize that. And I think there are certainly lessons. One of the lessons is that life in some ways, the fact that life has persisted for 4 billion years suggests that life is robust. On the other hand, individual species can be fragile. And we do see at times in the past that, when climate changes rapidly, it's generally bad for biological diversity. And in fact, one of the great mass extinctions 250 million years ago, we think was driven by volcanism 1 million times larger than anything ever experienced by humans. And interestingly, the direct consequences of that are global warming, ocean acidification, a decrease in oxygen in the subsurface waters of the ocean. And if you read the newspaper, those are pretty familiar phenomena. It's exactly what's happening today. And some groups of organisms fared pretty well at the end of the Permian. Others were obliterated. And it turns out that the pattern that we saw in the Permian does a good job of predicting vulnerabilities of things today. So I think there are those lessons. I'm not sure, on the other hand, that the solutions to present-day climate change come from those. I think what they suggest is that we really need to be serious about finding solutions, which is why I think many of us are pleased at that first step that was taken in Paris last week. But I think the solutions will come from the political will of populations to live our lives in a different way, and by technology that will both be much more efficient in the use of energy, sources of renewable energies that don't create CO2 as a byproduct. And I suspect technologies which exist now but are very, very expensive to actually actively remove CO2 from the atmosphere. And I suspect over the next 50 years, those are going to be very prominent lines of interest for everyone in the planet. There's another question from this lady here.
Professor, do you believe that this increase in human population in the last 200 years will be a problem that we will face, and will it affect the environment and the earth in general as it keeps increasing?
Yes. Yeah, the question is what about the human population? And again, it's a two-sided coin. In a sense, there's no question that the population of the world, which, at the time of Christopher Columbus was maybe 500 million or something like this, has increased dramatically to the point where we have more than 7 billion people now, and perhaps 2 more billion by late in this century. And in a sense, that reflects some of the greatest advances in science and technology over the 20th century. I think, in some ways, we all live in this golden age for humanity because the things that kept populations down, diseases, poverty, and that, have started to be addressed in ways that allow more people to survive and reproduce. And so you would have to be very grumpy to think that it's bad that we've made life safer for much of the world's population. That said, almost all the things that we do worry about for the 20th century are a direct function of having 7 billion humans. And so I think that one of the challenges is how do we feed not only 7 billion people, but how do we feed 9 billion people? It's been estimated that we'll have to increase global agricultural production by 20 or 30% over the next 50 years to feed the population. How do we do that without causing more offense to the environment? And people, very good people are trying to work on solutions to this, working on solutions to family size management, working on solutions to energy issues. And I think the real question and the challenge for everyone is how do we make the changes that we need to make to bring ourselves into some sort of sustainable alignment with the environment, but still allow people in developing nations to have economic aspirations. It's not a very good solution if we in the US or you in Sweden say, well, it's all right, we'll just keep things as we are now, and people in India can remain poor and will remain prosperous. I think we have to have solutions that can actually increase the well-being of people around the world. And again, I think the good news is that most people in the world now take that challenge seriously, and there are very good, doctors, scientists, engineers all over the world who are working on these. And I think for those of you who may wish to become scientists or engineers, there's no bigger challenge to your generation than to contribute to the world that we all hope we can find over the next 50 years or so. What else?
So I can ask a question.
Oh, no, you can't.
So you've worked in some pretty exotic places. You've worked in Greenland, Svalbard, in Siberia.
Yeah.
And they're pretty cold places, especially someone coming from more the equator. Yeah. I have 3 questions for you. Did you go there voluntarily, or did they make you? Why would anyone ever want to go to Siberia? And what were the important discoveries you made while you were there?
Well, I have to say that once when I was in Siberia, we actually came to the village where Stalin was imprisoned by the Tsar, and it was a terrific place to put Stalin. The mistake turns out to be letting him out in the 1920s. Yes, I mean, in a sense, they're exotic places, but they're actually wonderful places because there are no humans there. So I've spent a lot of my time in Spitsbergen, at the northern end of Scandinavia. It's like having the Alps at sea level, beautiful rocks. The rocks are exposed spectacularly well. There are polar bears, but you learn to live with that. And they're just wonderful places because the geologic record is exposed beautifully. And the same goes for northern Siberia, where we work is essentially uninhabited ecosystems. They're quite beautiful. You float down these rivers in rafts and look at rocks exposed in cliffs. And again, these are, I opened by saying that sedimentary rocks were libraries, and you have to go to the right library to answer questions. And so the reason we went to Siberia is that there are 2 sets of rocks exposed in the wilderness of northern Siberia that help us to understand Earth's middle age, that interval of low oxygen, and also some that help us to understand the rise of animals, this whole sequence of biological and environmental changes as we move into the modern world. And Siberia turns out to have some of the best records for those. So no small part of what we know about Earth's middle age comes from work that our Russian colleagues and we did on the biological and physical characteristics of these rocks exposed in Siberia. And also much of the foundation of what we think we know about the so-called Neoproterozoic, that is the world from 800 million years to the dawn of animals, some of the best rocks in the world to ask questions about biological and physical changes in those worlds is in Spitsbergen. And there we found not only some of the best records of life of that age, but also we learned that the carbon cycle was working in a very different way. So when I was a boy, I used to sit and look at atlases on rainy days, and I remember I must have been 6 years old, and I said, someday I'm going to get to London. That was my great ambition. And being a geologist helps you get to London and a whole lot of other places too. So it's been very rewarding in that sense. Yes.
I also, I'll come back to you. I also think you're a little bit bipolar. And it's maybe it's you like working at both poles, but you also work in really hot places.
Sadly,
Namibia,
yes.
In the middle of Australia.
Well, it's good particularly if you work, and this is the way you can make the system work for you. If you work in Spitsbergen in the summer, you owe it to yourself to look at its modern sedimentological analogs in the winter, which are in the Bahamas and places like that. So there are some advantages of working here. I must say that I like working in dry tropical areas like the Namib Desert and the deserts of Australia, but I will say that the quality of preservation of rocks is much better in the Arctic than it is in these dry places. Australia, sadly, has not had an ice age in the last 300 million years, and it needs one desperately because all of its surface rocks are highly weathered. Whereas the rocks that we look at in Spitsbergen were beneath ice 15,000 years ago, and they're very pristine, which helps us in everything we do.
Yeah. Yes, talking about exotic places to be. I mean, what you have talked about is the history from a geo and bio perspective on the Earth, but it's hard to predict the future from the data. But we have got maybe the opportunity now, the last decades, by the exploration of Mars.
Yeah.
And that might be the future of the Earth, I don't know. But
Well,
what are your expectations there? What would you like to do type of experiments, and would you like to go there?
I don't want to go there. That's the first thing. I'll leave that to Matt Damon. But I think I actually stumbled into Mars research when a man named Steve Squires, who's a professor at Cornell University in the US, was building a proposal for a Mars mission. I think, wisely realized that you couldn't just have people who ran instruments, although you need people who can design and build chemical instruments and cameras that can actually function on the Martian surface. And these are really exceptionally talented people. But Steve said, we actually need some people who can interpret sedimentary rocks. And so he came up to me at a meeting and said, how'd you like to work on Mars? And I said, yeah, sure, why not? And it turned out the proposal was successful. So beginning in 2003, there were 2 rovers named Spirit and Opportunity that were launched to Mars, landed successfully. And so over the last 12 years, I've been fortunate to be part of a team which in a sense is doing on Mars just what we've been doing on Earth. That is, we have ancient sedimentary rocks which are exposed usually in the walls of craters on Mars, and we can look at their physical and chemical characteristics and try and understand the history of another planet. And there's a newer rover named Curiosity, for which my long-term field partner on the Earth, John Grotzinger, has been the principal scientist. And what we're learning about Mars is that it is certainly true that early in its history, Mars had more water, liquid water, than we see at present. Liquid water is in fact not stable on the Martian surface because the atmospheric pressure is so low that essentially ice sublimes right to water vapor. It's near the triple point of water. So I don't think that present day life is a very likely possibility on Mars, but it's at least possible that 3 or 4 billion years ago, Mars had the physical ingredients that might have given rise to Mars. Now, having said that, one of the records that water leaves on Mars is just in the minerals that are deposited when the water evaporates. So just like when you take seawater and evaporate it here on Earth, you will get gypsum and table salt and other more exotic salts. We have those salts from the evaporation of water on Mars. And 2 of the things that our group has worked on is, what do they tell you about environments on Mars? And they suggest that even when you had water, that water would evaporate in a way that would make the water too salty for any life that we understand on Earth. And also, it's very interesting that at least some of the minerals that were formed by interaction with water early in Martian history, some sulfate minerals, some clays, some silica for Daniel. These things are all minerals that don't last over multi-million year timescales on Earth because the minerals transform to other substances in the presence of water. So what we're learning on Mars is that some of these things that formed deep in the past on Mars in the presence of water have not seen appreciable water since they formed. So from that, I would take it that Mars may have been somewhat different early in its history, but it was, broadly similar to today for, much of the last 2 billion years or so. Whether or not, I don't think there's a particularly helpful lesson for our future. I don't think we're in danger of running out of water. But I am always taken that in some ways, people looking at Mars came up with the idea of directional planetary history before people working on Earth did, and we can certainly take that lesson from Mars. What else? Anything else? Yeah, there's one in there. Yes.
Okay, so on this graph you're showing time in that direction, but the previous slide
yes.
you had time that direction. Is there ever a reason for that?
It's individual taste. I think geologists tend to be linear and go from the past on the left to the present on the right. Biologists, for some reason, like to go from what we know best to what we know least. But there's no real convention. Yeah, sorry. I'll take it up with my geologist friends. So. What else? Anything else?
Stefan. Okay, there's...
Oh, okay. There's a...
Okay, so what's the answer to the question that Earth can teach us about the question of all questions, namely, are we alone in the universe?
That's another one of these questions that if I knew the answer, I would write about it. The question is, what does the Earth tell us about possibly the distribution of life elsewhere? And in some ways, the answer is the most obvious thing, that Earth turns out to be the only place in the universe that we know life exists. So that we have some confidence that at least on planets that are physically broadly similar to the Earth, there might be a possibility of life. Now, before I go into that more, let me say that it is not obvious that the type of life that we see on Earth is necessarily the only type of life imaginable. One might imagine other types of molecules that could be informational or functional in that. But that becomes a game without rules very quickly. So if we at least use the one life we know of as our paradigm for life, we can ask about what would be the conditions on an early planet that might give rise to life. And certainly the combination of water, various solar and hydrothermal energy sources, and the absence of oxygen on Earth are all the kinds of conditions that many people think would have incubated the chemistry that led to self-replicating and evolving systems. So then the question becomes, if you think that's what you should look for, other Earth-like planets, how do you do it? And it turns out, over the entirety of the universe, there's 3 ways that you might look for life or Earth-like planets elsewhere. First of all, within our solar system, we can explore. We can actually go to Mars. We can go to the moons of Jupiter and Saturn, some of which may be the other most biologically interesting objects in our solar system. And that will be done. It's being done now. It will increase over the next few decades. So I think that, by the time most people in the audience are your age, we will probably know with some confidence whether or not life is unique to the Earth in this solar system. If I were a betting man, I would bet that it probably is, but it's possible we could all be surprised. The harder part has been going beyond our solar system to look for planets around other stars. And that, as many of you know, has been something that has just mushroomed as an observational science just in the last few years. The first claim of an extrasolar planet, that is a planet around another star outside our solar system, was only made in the 1970s. And today there are thousands of known stars with extrasolar planets around them. The trick has been to find planets the size of the Earth, because the way you actually look for planets is to look at, as a planet revolves around its sun, there's an influence, the planet and the sun influence each other in terms of gravity. And if you're lucky enough to be in the right plane, when the planet goes in front of the sun, it actually occludes some of the light from the sun. And even though that might be 1 part in a million, instruments can actually read that. And so as you can imagine, the first planets that were found along other star systems were planets the size of Jupiter that revolved around their planet every 3 days, because those are big planets that have fast or short periods, so you can actually see them. Now it's becoming clear that most planets aren't giant planets. As we've looked with more powerful instruments over longer time periods, we're finding that planets between the size of Earth and Neptune, usually called super-Earths, are probably the majority of the planets. So just in the past year or two, people have suggested that they've discovered planets that are, at least in their physical dimensions, similar to Earth. Now, the ones that are known are actually near enough to their stars that you probably don't have liquid water there. But I think sooner or later we will find planets that fit this Goldilocks position of being at just the right position relative to their stars. Then the question becomes, what do you do about it for biology? And your best bet is, just as we live in an oxygen-rich planet on Earth because of photosynthesis, one will look for signatures in the atmospheres of these extrasolar planets and see if you can make some biological inferences for them. And then finally, that takes us just a few light years away from Earth. The rest of the universe, to be honest, I think the only way we find out about life in the rest of the universe is if they call us up and tell us about it. So, any other questions? Yep.
If life,
sorry, I don't know where you're. Oh, okay. Yep. Okay. Yep.
If life would develop on another planet, do you believe the life would, like an Earth-like planet with the same conditions, do you believe it would look similar with cells as on Earth, or would it be completely different?
Yeah, that's a really good question. The question is, if we do find another example of life, will we recognize it? Will it be like us? And I think there would be some things that I think would be similar and other possible differences. I think the things most likely to be similar are actually the things that in Earth history developed first. That is, bacteria-like single-celled organisms. It turns out that if you look at the chemistry of how you actually make energy that's biologically useful, there are only so many reactions that you can use, and bacteria figured those out long ago. So if there's life elsewhere, I think it will be based on carbon, because carbon has a number of unusual features, including that it can be a solid or a gas or dissolved as ions in liquid at temperatures where liquid water is present. And also carbon has these wonderful binding possibilities. So there's something like 4,000 or 5,000 minerals that are known made of different elements, but there are hundreds of thousands of biochemicals, carbon carbon compounds known. And so I suspect that if we find life on the planet Remulon, it will be based on carbon. It will be based on the cycling of carbon and other biologically useful materials. Whether or not it has DNA, I don't know, but it will probably have some sort of informational molecule. Whether or not it has proteins, I don't know, but it will have molecules that actually give structure and function. And then it gets kind of interesting. If there was on American TV, there was a show not long ago which had some speculation on life on other planets, and they kind of look like elephants. And you think, well, we don't know whether. I mean, 100 million years ago, you would have never predicted elephants on our planet, let alone on another. So once you get, is it possible that there are multicellular organisms elsewhere? Yes, although they have to be obedient to the same rules of physics and chemistry. So that is, anything that exists on the planet Remulon is going to have the same relationship to physics that an elephant does walking across the African veldt. That doesn't mean that we would find it a familiar organism. And of course, the big question is there anybody else out there who can think? And the only thing we can say from our planet is that life started once and it got to us one way or another. And so we don't know whether that was a rare event. The reason we know it happened is that we're the product of it, and whether it's rare or common, we're here. As I said, we will. It's hard to even make probabilities about that, and so really, I think the thinking about, to my way of thinking, the best work we can do on the possibility of life elsewhere is just to observe. And to actually have space missions that will help us to at least constrain the possibilities. What else? Okay. One more.
So, Andy, I don't know about you, but I look out in the audience and I see a room full of future scientists. And you're one of the foremost scientists in our area. You actually work between geology and biology and chemistry. What kind of advice can you give these young scientists to be?
Well, first of all, I think there are a lot of opportunities in science. I know that some of you will become businessmen, and if you do, you could do like the Crawfords and do good for science through philanthropy, which I think is wonderful. If you're going to become a scientist, I think that the best advice I can give is to love what you do. If you don't get up in the morning really wanting to solve problems, in my case, really wanting to know how the Earth works, you won't be happy. So you have to be, like any other, I think, profession, it helps to love what you're doing. As far as anything more direct than that, I really do think that, the 20th century gave us physics and chemistry and biology and geology and astronomy, and often these were in different departments that were peopled by professors who hardly talked to each other. And in 1950 or 60, the way to be successful in science was to find a narrow discipline and bore as deeply as you can down that hole. I think in the 21st century, the most interesting and I think most important from the standpoint of both fundamental questions and societal questions, the most interesting science is being done, as I say, at the gaps between the traditional things. So chemistry and physics come together in interesting places. As we've seen today, biology and geology come together in interesting ways. And I think more and more the sort of departmental boundaries that Daniel and I grew up with are going to seem antiquated. And most of the science is going to be multidisciplinary efforts built around questions. So what that turns out to be, as far as advice, is just A, be curious, and be curious about everything around you, and learn, physics, chemistry, and biology, because no matter which of these, or if you're in earth sciences or astronomy, you're going to need to be able to talk to people in all of these areas. So learn everything is the only question.
Okay, before we end here, I'm going to turn the word over to Stefan Bengtson. No? No? Ebba, I'm sorry.
It's okay.
Sorry.
Thank you. My name is Ebba Fischer. It's a very strong light here. I'm the chair of the Crafoord Foundation here in Lund. And Holger and Anna-Greta Crafoord on the golden medal there were my grandparents. And I'm so honored and proud to have you here, Andrew. As Stefan told us before, the wish and the purpose from my grandfather when he for the Crafoord Foundation was to support science, scientists, and the education of young people. So today, of course, he's sitting up there smiling when all of you here. And I hope this afternoon has inspired you to be even more curious about working with science in the future, because you are the future. And once again, Andrew, I'm so happy that we could make this together, together with the Royal Swedish Academy of Science, of course. And here we have a diploma to you,
Oh, thank you.
to thank you for this
Thank you very
afternoon.
much.